I won't let you poison this body! Kamehameha!
Okay, this video is going to be about berries, such as strawberries, blueberries, raspberries, and their multiple effects on different aspects of cancer. Also, having to do with the effect of a PMAC and a CMAC, which is what the tumor fears and hates, because the PMAC is the end of the tumor. Okay, so these berries are strong antioxidants, also membrane stabilizing effects. That's important for adhesion machinery, for things like PMAC, CD receptors for immune cells. Also, membrane stability is important for the prevention of things leaking out of the membranes if the membranes are protecting the cells, thanks to berries. Also, I'm going to be talking about berries and the effects on lysosomal function. Okay, so berries have a lot of, let's say, anthocyanins like cyanidin, delphinidin, flavonoids like quercetin, which is critical to avoid excessive zinc, because excessive zinc could raise testosterone levels too much, and then that could lower estrogen, which will also lower magnesium levels, which are critical for the lysosome, because the low estrogen levels also means less magnesium transporters, and that means less magnesium, and then lysosomal membrane peroxidation, and then other issues with the lysosome being dysfunctional. But so berries help with that thanks to the quercetin. Okay, so the antioxidants in berries are critical for lysosomal function and fusion. So berries reduce reactive oxygen species that preserves lysosomal membrane integrity. Also, berries limit lipid peroxidation, stabilizing SNARE dependent fusion machinery. Okay, now that's important so that the lysosome loaded up with tumor exosomes that could go to the tumors, which are the exosomes going to the tumor, could end up happening if the MVB fuses to the plasma membrane. But that can be avoided thanks to berries because it increases lysosomal function, protects the lysosomal membrane, and the berries protects the snare machinery that is supposed to prevent the exosomes from going to the tumors. And the way it does that is that the snare machinery which the berries protects allows the MVBs holding the exosomes to fuse to the lysosomal membrane instead of the plasma membrane. If the MVB sacs or the exosomes fuses to the lysosomal membrane, then the tumors will not get the exosomes. And that's possible, of course, if berries protects the snare machinery that is required for the MVB sacs to fuse to the lysosome. Protect the lysosomal membrane from leaking out, to stop the cathepsins from leaking, because the cathepsins are needed for the lysosome to degrade the exosomes. Next is berries also lower oxidative cholesterol conversion, therefore leading to less oxysterol accumulation. That's important to reduce the oxysterols because the oxysterols, they load into the exosomes or actually form the exosomes themselves. High cholesterol can also cause the lysosome to be dysfunctional, and the lysosome cannot degrade the exosomes, causing the tumors to get the exosomes. But thanks to berries, berries lower and reduce cholesterol, and enhancing and enabling lysosomal function. The next benefit of berries is critically important for PMAC formation, which is what tumors hate because it is pretty much the end of the tumor, and that is berries, by reducing reactive oxygen species, increases talon unfolding domains and actin filament dynamics, because the PMAC formation relies on actin. So berries, with their very strong and high antioxidant capacity, protects the integrin, talon kindling, actin integrin clutch system, which is required for the PMAC formation. This is also critical because it biases cells toward adhesion stability, therefore allowing like adhesion molecules and machinery that helps hold and tether CD receptors onto immune cells. The functions will have to be explained in a different video. Also, this also prevents apoptosis, possibly induced by tumors. So berries, they also help improve and form what tumors hate and fear, which is the PMAC. Because the berries, they reduce oxidative misfolding, because berries have a very strong antioxidant capability. Therefore, sustained PMAC signaling competence. Also, berries, they stabilize lipid rafts, which improves TCR integrin microclustering. That's important for the actin integrin clutch that the PMAC relies on to be able to form. Berries also lead to stabilized lipid rafts. That's important to prevent CD70 on the tumor, because CD70 will oppress CD27 on the immune cell, ongoing NFKB, and then exhausted T cells. That situation is very hard to reverse as well, but it is possible, but it's better to avoid it. So berries, they reduce reactive oxygen species, prevents cholesterol, therefore saving the lysosomal membrane. This is consistent with the better receptor clustering. That's important on the immune cells, because immune cells, they need receptor clustering to be able to express their receptors, because immune cells, they have a CD25 receptor that takes up IL-2, because the T cells need IL-2 to be able to function, or else the T cell will be weak. But the CD25 has to be tethered and expressing on the surface of the T cell, which is what berries help with a better receptor clustering. Okay, now I'm just going to touch more on the receptor clustering part. So berries contain polyphenols that they insert into and interact with the lipid bilayer to reduce membrane oxidative damage. That way, the CD25 receptor for the T cell can tether on to the membrane, because the berries increase the membrane rigidity, so that the CD25 receptor even has anywhere to tether onto. This matters because membrane fluidity affects how receptors move and cluster and signal. And the last benefit, but not the least, is berries reduces NS-MACE2, or NSMASE2. So compounds in berries downregulate that activity. What that does is that it reduces ceramide overproduction, because exosomes, they need ceramides. Tumors actually secrete ceramides to make exosomes. So less ceramides means less exosomes, and no exosomes for tumors. So that's it for the video. Like, comment, share, subscribe. Keep it locked in this channel, and I'll see you in the next video.